Imagine a world in which woolly mammoths roam the plains of Siberia, where dodos flock on the shores of Mauritius, or where 12-foot-tall, saber-toothed cats stroll through the woods of North America. Well, maybe no thanks to that last one. But a world in which extinct animals are brought back to life may not be limited to the imagination for much longer. De-extinction is the process of bringing back to life an extinct animal species. It's an idea that's been around for a while, but was perhaps fully propelled into the public consciousness by the 1993 film Jurassic Park. While resurrected dinosaurs remain firmly in the realms of science fiction, the de-extinction of other animals is a much closer horizon. We are currently living through Earth's sixth mass extinction period, called the Holocene extinction. This mass extinction closely correlates to human activity. Through hunting, deforestation, pollution, climate change, and other side effects of human expansion, we may have lost close to 7% of all species on the planet. But de-extinction could be one way of resurrecting lost species and protecting those most at risk today. And with recent developments in genome sequencing, genetic engineering, and cloning, scientists have now begun to resurrect recently extinct species and have their sights set on those lost further back in time. So, is a Jurassic Park-style zoo filled with extinct species actually possible? What would we need to do to bring back animals that would otherwise be lost forever? To fill our hypothetical zoo, one of the first decisions would be which animals to bring back to life. This DNA is either non-existent or only present in tiny amounts. So, for better or worse, that rules out the dinosaurs. It's also much harder to de-extinct birds or reptiles compared to mammals because we know so much more about mammalian reproduction compared to birds and reptiles. But if we had a mammal in mind which lived within the last few thousand years, how would we go about bringing them back? There are three main routes currently being explored in the push to de-extinct animals, and they all have pros and cons. If we had lots of time and a few acres of land, we could try backbreeding. Backbreeding is a process where lost traits are bred back into living species, sort of converting an existing species back into a previous form. To understand this method, let's look at an attempt by researchers in South Africa to recreate the quagga, an extinct subspecies of zebra. Quaggas and zebras are pretty similar, but where zebras are covered in stripes, quaggas only have stripes on the front half of their bodies. Quaggas also have brown coloring on their rear. Quaggas were common in South Africa until European settlers hunted them into extinction in the late 19th century. Because zebras are a closely related species to the extinct quagga, they are the starting point of the study. To begin recreating the quagga, scientists select individual zebras that express the same or very similar traits to the extinct species, such as zebras with fewer stripes on the back half of their bodies, or zebras with more brown coloring on their back half. Then, once these individual animals have been identified, they are bred together. Any offspring that have inherited the desired traits will be bred again, hopefully producing a new generation that carry a mix of extinct species traits. This process needs to be repeated until the resulting animal is as close to the extinct species as possible. The backbreeding project started over 30 years ago, and in that time has produced multiple breeding groups of nearly quaggas. After several generations of this selective breeding, the animals aren't quite the same as the originals, but they do have the half stripes and brown rears of their extinct relatives. But there are some downsides and challenges to backbreeding that mean it might not be a suitable approach for every animal. First of all, we'd need a really close relative of the extinct species to act as our starting point. For animals extinct many thousands of years ago, this may not be possible. It's also a really long process. The gestation period for a zebra, for example, is 12 months. If we need to repeat the breeding process several times over, it could be years or even decades before a herd of our extinct species is ready. And it's an imprecise process. Although genome sequencing and in vitro fertilization can help breeders be more selective about what gets passed on, you can't guarantee the traits you expect to see in the new generation will materialize. But luckily, even if we don't have a starter species, or we don't have time to breed multiple generations, there are some other options. Another route to de-extinction is cloning. Clones are exact genetic replicas of another organism, and arise all the time in nature. Bacteria clone themselves as a means of reproduction, and identical twins are technically clones of each other. But cloning can also be done in the lab. We've cloned lots of animals, including buffalo, dogs, and camels. So how could we use cloning to bring back a long-dead species? The most reliable method is probably SCNT, somatic cell nuclear transfer. This is the method pioneered by the creators of Dolly the sheep, the first cloned mammal. And surprisingly, scientists have already attempted this method. It began in 1999, when researchers captured the last surviving Pyrenean Ibex, a female named Celia. They obtained skin biopsies and froze the tissue in liquid nitrogen. Then, in 2009, those skin cells were used to clone Celia, in the first ever case of actual de-extinction. First, the nuclei from the frozen skin cells were removed and injected into donor eggs taken from domestic goats, which had their own nuclei removed. By adding some chemicals and a jolt of electricity, the process of embryonic development was started. After a few days of growth in the lab, the embryos were implanted into a surrogate mother, either a Spanish ibex or an ibex goat hybrid. The resulting fetuses had exactly the same DNA as the extinct animal. Of the 208 embryos the researchers implanted, only 7 pregnancies resulted, and just 1 ibex made it to term. This was a huge accomplishment, the first ever extinct animal brought back into existence. However, the baby ibex died almost immediately from respiratory failure. Most embryos derived from clones don't develop properly, and we don't fully know why. Research suggests that it's not just one stage of the cloning process that is causing damage to the resulting embryos, but in fact many, if not all stages contribute. From the act of moving the nucleus from one cell to another, to the use of chemicals to kickstart embryonic development. But scientists are not ready to give up so quickly, instead setting their sights on an even loftier goal: de-extincting one of the greatest megafauna of all time, the woolly mammoth. In 2019, a major breakthrough in the effort to bring back this incredible animal occurred, when a team in Japan successfully transferred the nucleus from a frozen mammoth cell into the egg of a mouse. The nucleus came from a 28,000-year-old preserved mammoth named Yucca. The goal of the experiment was not to create some kind of mammoth-mouse hybrid. It was to see if this ancient DNA had the potential to be reawakened. Could the cellular machinery in a mouse egg fix the damaged parts of the mammoth DNA and kickstart the DNA replication necessary for embryonic development? If it could, that would suggest there is potential for harvested, preserved mammoth DNA to be brought back to life through cloning. And the mouse cell machinery did manage to fix some parts of the damaged DNA, and there were some very early signs of the mechanics that underpin DNA replication. But ultimately, the mouse egg could only do so much. The mammoth DNA was too badly damaged, and so it's nearly impossible to reconstitute it enough to actually bring it back to life. For now, cloning a mammoth is still a far-off dream, but research like this adds to the field of knowledge that might get us there one day. But there is one other approach we could try, and it has elements of backbreeding and cloning. A new technology opening up options for de-extinction is genetic engineering, more specifically CRISPR technology. CRISPR-Cas9 is a type of molecular scissor that can cut DNA at a pre-programmed position. This means strands of DNA can be added, removed, or edited at specific points in the DNA. We could insert genes from an extinct animal into the DNA of a close relative, creating a hybrid with the phenotype, the physical attributes of the extinct species. To do this, we'd first need to sequence the extinct animal genome. If we have preserved DNA, for example from mammoth tissue in the permafrost, we can use genome sequencing to work out its entire genetic code, and use tests to work out which genes are linked to the phenotype of the extinct species. If we then compare the extinct species' genetic code to that of its closest living relative, we could work out where they differ. Finally, we could copy and paste the genes we want from the existing animal into the host animal DNA. Researchers at Harvard University are trying to use this CRISPR technology to splice mammoth genes into Asian elephant DNA. But it's a monumental task. There are around 1.4 million known gene mutations separating mammoths and Asian elephants. And not all of the mutations will relate to the mammoth phenotype. It will take a long while yet to work out which mammoth genes need to be swapped into elephant DNA to recreate a woolly mammoth. But the idea may be the most promising yet, and one day we may just see the first baby mammoth to live in 10,000 years. Although none of these methods are perfect, they are certainly bringing us closer to the de-extinction of lost species, and to protecting and reviving endangered animal populations. But before we get too excited about bringing back everything from mammoths to thylacines to dodos, we might want to pause and decide whether we should bring back any lost species at all. Because as cool as it might be to create a de-extinction safari park, there are some risks. While reintroducing recently extinct species might prove beneficial to existing ecosystems, long-dead species could end up doing more harm than good. Because of the changes those ecosystems have undergone in the intervening period, it could create similar problems to those we've seen when releasing non-native species into new areas. When the poisonous cane toad was introduced to Australia to control the grey-backed cane beetle, the toad quickly spread and pushed out native toad populations. An extinct species might outcompete one that has developed in its absence. Or the ecosystems that once supported an extinct species might no longer be a suitable home. The Chinese river dolphin was polluted out of the Yangtze River, and things haven't improved since. They wouldn't survive there today. If and when we are able to successfully and reliably bring back lost species, particularly those that have been extinct for thousands of years, we will need to think carefully about whether these are risks we want to take. But every day, between 30 and 150 species of animal die out on Earth for good. Alongside conservation efforts, de-extinction could be a potential avenue to undo some of the damage that humans have enacted on the planet. One of the most mysterious and strange animals to ever live on our planet was the thylacine, found in Australia, Tasmania, and New Guinea. It was the largest carnivorous marsupial, with a body that looks like a cross between a cat and a dog. Also known as the Tasmanian tiger, it was wiped from the continent of Australia 3,000 years ago, and survived in Tasmania until the 1930s, when humans finally hunted it into extinction. Over recent years, there have been many reports of thylacine sightings in the jungles of Tasmania, giving some hope that a few individuals remain. But these sightings are almost all exaggerated or are cases of mistaken identity. Sadly, the thylacine is still very much extinct. But it may not be forever. Geneticists are working to resurrect this enigmatic animal with CRISPR and cloning, working with a tiny relative of the thylacine, a numbat, to see how scientists are working to understand these marsupial genetics and to see how close they are to making a living thylacine a reality. You should watch Resurrecting Tasmanian Tigers on CuriosityStream. CuriosityStream is a streaming platform with thousands of high-quality documentaries like this one.